Okay, so what I want to do now is to introduce an online tool to you, which is called CADIMA, that was developed here at JKI. And furthermore, I want to very briefly summarize the results of a review that was conducted in order to identify and also to characterize online tools that are currently on the market. So before starting, I want to also provide you with a small brief introduction into the JKI, so the Julius Kühne Institute I'm working for. So the JKI is the Federal Research Center for Cultivated Plants in Germany, and it's both a research institute and also a higher federal authority. And the major tasks of JKI are threefold. So we are performing own research activities in order to support our scientific assessment and also to support the provision of policy advice we have to provide for the German Ministry of Food and Agriculture. And the research activities, they do cover the cultivated plant in a very holistic approach. So they tackle plant genetics, breeding research, plant nutrition, agronomy, soil sciences, and also plant protection and plant health. So within the JKI, I'm working for the Institute for Biosafety and Plant Biotechnology. And the scope of this institute is the impact assessment, which includes both risks and benefits of plant biotechnology, and it further includes the classical genetic engineering, but also new plant breeding techniques. And so how we now stumbled over the evidence synthesis approaches, and this happened in the course of a recently finalized EU-funded project, which is called GRACE. And in the course of this project, systematic reviews and maps were performed on potential impacts of genetically modified plants on human animal health, socioeconomy, and also on the environment. And in the course of this project, it was decided that an open access tool should be established that supports these synthesis activities, and this tool was named CADIMA. And very importantly, CADIMA will be permanently maintained and further developed by JKI. So we have the commitment by the President of JKI and also by the Ministry of Food and Agriculture. Also important to mention is that CADIMA was developed in a very close collaboration between JKI and the Collaboration for Environmental Evidence. Okay, in the following I want to very briefly introduce CADIMA to you. So if you want to work with this tool, you have to go to the webpage, so www.cadima.info, and on the main settings page you are free to mention or to lay down the title of your review, the synthesis approach you want to follow. You can specify the question type you want to address, and you can customize your review team. And very importantly, if you look at the menu structure, you can see that it very much resembles the core steps of a systematic review. And so in the following, I now want to very briefly introduce to you the core things that are possible with CADIMA. So I want to skip now the protocol stage and start with the literature search. So very importantly, CADIMA is not a search engine like Web of Science, but what you can do is that you can exchange reference lists, for example, with endnotes as RIS files. And the purpose is that you do allocate a search string that you apply to a database that you applied it. You have to mention when you applied it, and you have to upload your search results. In the next step, duplicates are recognized within CADIMA, and if the review team wish so, and they are also automatically removed. And what is planned now in the next step is that CADIMA will allow for a sorting of records considering the likely relevance for the question, meaning that records which are likely more relevant for the question will come first, can be rated first, and relevant references that are most likely not that relevant for the question will come at the very end. The next step would then be the study selection step, and at this stage you can decide the application mode, meaning if you want to screen title abstracts together and then full text, or if you first want to screen title followed by abstract followed by full text. Furthermore, you can decide if you want to do the selection process independently by more than one reviewer or not, or if you decide that part of the records might be screened independently by two, you can also allocate this by using these sliders in order to allocate the records to the review team members. So very importantly, currently the same reviewers will be involved at title, abstract and full text stage, but we are working on an update where we are absolutely free in allocating different records to different reviewers at title, abstract and full text stage as well. Furthermore, CADIMA does support the automatic calculation and also the reporting of consistency check results. So currently provides a kappa value, but also an Excel file, which provides you with the different ratings if you wish to calculate other values than just relying on kappa. Now coming to the screening step, how it looks like in CADIMA. So we're now at title abstract stage. So on the left hand side you have here the criteria you developed, and on the right hand side you have the abstract and the title, and then you can very easily go through the criteria and check if this title or this record seems to be relevant or not at title abstract stage. Very importantly, if you do it independently by more than one reviewer, CADIMA also automatically recognizes discrepancies in the ratings between the reviewers that are involved, and you are forced to solve these discrepancies. Very similar at the full text stage. So here you have on the left hand side the criteria, on the right hand side the PDF. Again, discrepancy will be highlighted in case it is done by more than one reviewer independently. And also what CADIMA is supporting is a bulk upload of PDFs, meaning that you do not have to upload one PDF by the other, but you can just do a bulk upload and CADIMA automatically allocates the different PDFs to the right entries. The next step would now be the data extraction and of extract or included studies, and CADIMA at this stage does allow for both an online and also for an offline data extraction. And if you now go to this in CADIMA, you can see in the top how it looks like. So you have your extracted studies, you can click on them, and then you can online type in the values that you want to extract. In the case that you want to do it offline, you can download an Excel sheet, you can compile the Excel sheet at your desktop at home, and this Excel sheet you can then re-upload to CADIMA, and CADIMA puts the different variables into the right places. What we are working on for the future is that you can also use code sets in the form of drop-down lists, so that you can define which values might be used when extracting the data in order to be more harmonized, let's say, in the extraction mode between the different or the review team members that are involved in this process. And very importantly at this stage, you can also determine which variables are relevant for the critical appraisal, and they are here marked in orange. And this you can do because if you're now going to the next stage, which is the critical appraisal of included studies, only those variables will be displayed, meaning that you do not have to go through your entire data extraction sheet, but only the relevant variables that you really need will be displayed, and like this the critical appraisal is facilitated. The critical appraisal in the next step, so I do not show it here, but it looks very similar to the selection process, that you can define your critical appraisal criteria, and then you, as already said, the variables are co-displayed, and then you can rate them or determine the tendency that they might be prone to a specific bias or not. Okay. The next thing is then the documentation. So what CADIMA is also doing that is it assures for thorough documentation. So it provides you with a flow diagram following the PRISMA requirements. It provides you with the results of your literature search, reference lists as Excel and also RIS files. So for example, you can then just use the RIS file that you get out of CADIMA. After the full text screening stage, you can, while you are writing up your review, you can upload it to EndNote or any further reference management software, and you can just use this in writing up your review. You get a list of selection criteria, You get the results of your consistency check, the outcomes of your study selection, the included references after title abstract and full text screening, as well as a list of your critical appraisal criteria, and the compiled data extraction sheet, and also the outcomes of your critical appraisal. So very importantly, CADIMA is not supporting the data synthesis step, as we decided that depending on the data structure, what you get out from a review, there are so many different tools, very good tools already available that you can use, and everybody is, let's say, familiarized with different tools. So we thought, okay, this we leave to the user, that he uses the tool that he's used to in order to synthesize the actual data. Furthermore, what CADIMA is also allowing for flexibility, meaning that you are not forced, like it was depicted now, that you have to go through the systematic review process in a step-by-step manner, but you can start one step and work on it, and then, for example, now you are absolutely free in adding new references and work with them, so you do not have to strictly adhere to the different steps. Okay, now I'm coming to the review of existing tools. And this part was done by Emma McIntosh, who did a fantastic job on this. And what she did is that she screened the literature and also databases or web pages, and identified and also tested 22 tools that do support the review process. And what you can see here is that the majority of the tools are either stick or tackling healthcare issues, or they are, which is very nice, so they are actually open to any kind of review question that you want to address. And a further issue, which is really nice, is that around 70% of the identified tools are actually open access. And so the tools that were identified and tested are depicted here. What Emma did is that then we defined some criteria, and she checked them if they do support the different criteria or not, and the results are very briefly summarized here. And so I do not want to go into detail, but what is clear is that there are many tools that actually do differ in the things, in the support they do provide for the review process. And as I guess almost every review has different demands, and the review team has different requests, I mean, it's up to the review team in order to check which of the tools is the most appropriate in order to support the evidence synthesis activities for these respective questions. And if you are interested to read more about this review, but also maybe on CADIMA, here are some 2 papers that we recently published in environmental evidence, and so please go and check and read further. Now, coming to my last slide, it's the acknowledgements. So the association people I want to thank, and especially I also want to thank AVM, as they made it possible for me to present now here in the course of this special session. And yes, thank you very much.